Hey guys, in this video we're going to be talking about some Mendelian genetics practice, which is basically simple genetics, and we're going to be talking about Punnett squares. All right, so let's go through a couple practice problems together and kind of go through some of these big ideas. All right, so our first practice problem here has to do with our rabbits. It says if the allele for brown coat color is dominant to the allele for white color in rabbits, what are the possible genotypes for the brown rabbits? All right, so it says we're going to use here dominant is a capital B. All right, so our first rabbit here is a brown rabbit, and we need to figure out its possible genotypes. But we know that as a brown rabbit, it has to have a capital B because that is the allele for brown coats. Now, we know a genotype is made of two alleles, right? So one of the two alleles has to be a capital B, but the other one could be this. So that is a brown rabbit, capital B, capital B. The other possibility we could have is a capital B and a lowercase b. Now this is still a brown rabbit because the capital B brown is dominant over the lowercase b, which stands for white. So as a phenotype, brown always shows over white if brown is there. So these are the two possibilities that we could have for a brown rabbit. Now let's take a look at the genotype for a white rabbit. A white rabbit, on the other hand, we know its allele has to be a small b. And if it has any large b's, it would not be white. It would be brown. So we know that it has to be lowercase b, or small b, homozygous recessive in order to have a white rabbit. Now let's try another example. Here we're talking about skin pigment, okay? And it says here that having skin pigment is a dominant trait. It's a big P. And if you do not have skin pigment, it's considered to be a lowercase p. It is recessive. All right, so let's try to figure out the phenotypes or what they would look like for each of these. All right, so here we have a heterozygous one, but one of them is a large P, so that means this person would have pigment. Now, let's try this next one here. We have a homozygous dominant. We know that dominant is pigment, so the phenotype for this individual would be pigment. Now this one here, we have homozygous recessive. This individual here, we would say is albino or has no pigment in them. All right, let's try this with a Punnett square. A Punnett square is used to predict what the offspring will look like if we cross two parents. So our first parent here is a heterozygous tall. We remember the word heterozygous means different, so it has two different alleles. So we're going to cross that with a short plant, and short is recessive. So the only way a plant can be recessive and the only way a plant can be short is if it is two recessive on it. All right, so what we do now is we set the parents up in our Punnett square. We separate the alleles. Okay, good. The large T gets distributed straight down. The lowercase t gets distributed straight down. And then the same thing happens for our other t's. They get distributed straight to the right. So here's our possible offspring. Okay, if we take a look at this, guys, if we would describe the genotypes, we could have either a heterozygous or a homozygous recessive. So 2 out of 4 are heterozygous, that's 50%, and 2 out of 4 are considered to be homozygous recessive, that's also 50%. If I would describe the phenotypes, we would have 2 tall, which is 50% tall, or 2 short, which is 50% short. Now, sometimes we like to move traits, genes, and we like to move them together or separate from each other and see how that would look. So in this situation here, we're going to be doing a dihybrid cross, which is a multiple step. But really all you're responsible for is knowing how to distribute. So for example, the parent here on the top would send down a little y and a big T. And the parent here on the left would send over a little y and a big T. That's it. Make sure you guys rewatch this. Make sure you understand how Punnett squares work.